Hello, my name is Carol Leibel, and today we want to examine the structure of cells. The cell theory says that all living things are composed of cells, and that the cell is the basic unit of life. Theodor Schwann and Jacob Schleiden in 1839 proposed those two things. Later, in 1855, Rudolf Virchow said that cells only come from pre-existing cells. And to this day, we have never found anything that is living that's an exception to the cell theory. There are basically two different types of cells. We have the prokaryotic cell, and a prokaryotic cell was the first cell to evolve, and these are cells without a proper nucleus or membrane-bound organelles. Larger, more sophisticated cells came into existence, and these are eukaryotic cells. They have a nucleus and membrane-bound organelles. We know that cells, for the most part, cannot be seen with the naked eye, and you have to use a microscope in order to observe them. So let's listen to this little video about cells.
Everything that lives is made of cells, even us. Although we can't see them, trillions of cells within our body work together to give us the gift of life. These tiny units of life fascinate biologists like Boyce Rensberger.
The entire human body is made out of cells, or the things the cells have made, such as bone. Skin is made of cells, your heart is made of cells, the muscles that operate the body so that you can move, these things are all made of cells, including the brain itself, which sends electrical signals around from one cell to the other so that we can think. All these different kinds of cells work fundamentally the same way. When you look inside the cell, you see the same kinds of structures.
Cells come in all sorts of sizes and shapes. They're so small that we need a microscope to see them. Inside of every eukaryotic cell is a microscopic factory that runs off chemical reactions. At the center, the nucleus acts as the cell's brain and controls its activities. It also holds the DNA, the instructions for life. The cell's membrane protects each cell and takes in the raw materials it needs to stay alive. Water, nutrients, and waste move into and out of the cell all the time. These materials may hitch a ride along the cell's transport network, a network made up of microtubules and intracellular membranes. The cell's transport network picks up, transports, and delivers material throughout the cell. The cell breaks down raw materials into the building blocks it needs to grow. This ability to take in vital materials allows the cell to function and perform specific tasks.
The instructions that are in the cells, starting in embryonic development, tell each cell how to change to take on a new function. Also, the cells monitor the activities going on. If there's a wound, the cells can sense that, and they will send out the message to other cells to come and repair the damage. So all of this keeps the whole body on a kind of an even keel.
Our cells work together in extraordinary ways. The heart, for example, is made up of muscle cells with a tendency to twitch. Separately, they twitch to their own rhythm, but together they beat as one. Millions of them work together to give us a heartbeat. These muscle cells survive as long as we do, but throughout the body, cells die and get replaced all the time. Our body produces new cells through mitosis, the process where one cell divides into two. Since we're constantly losing and replacing cells, most cells in our body are actually younger than we are.
So let's look at why cells are so small. We are looking at right here the size of cells. Prokaryotic cells are between 1 and 10 micrometers long. and eukaryotic cells are between 10 micrometers and 100 micrometers, which means that there's a factor of about 10 between the two. In order to observe cells, you have to use a microscope. There are a couple exceptions. For instance, an egg is technically only one cell. However, there's only one little area on the yolk that's metabolically active. The rest of the egg white, and the rest of the yolk, nothing is happening. It's inert material, if you will. A nerve cell is technically one cell, but they can be very long. It can go from your spine all the way to the end of your toe, and that's one cell, but they are extremely, extremely thin. So you might be asking, why are cells so small? Why can't you be composed of one nice large cell? Well, there are two reasons. The surface area increases at a much slower rate than the volume, and that is the volume of the cell if it were a sphere or a cube. And the second reason is the larger the cell, the longer it takes for the materials to diffuse to the center of the cell. And that you have to have adequate surface area to exchange materials with the environment. Alright, so let's look at an example of this. We have a cube here, and we'll say, hey, let's suppose that's one cell, and let's suppose that it has a unit of 1 for its length, width, and height. And the exact units are not important. And so if we were to determine the surface area, we know the surface area is the length times the width, and that there are 6 faces to the cube, and that it has a unit of 1. The volume is the height times the length, times the width, which is 1. So when you cancel this all out, this is going to be a 6 to 1, or the surface area to volume ratio is 6. Now let's increase the volume so that now the height and the width and the length is 5. So if you want to determine the surface area, again there are 6 faces to the cube. The length is 5 and the width is 5. The volume, we have the height is 5, the length is 5, and the width is 5. So that now the surface area to volume ratio is 6 to 5, or 1 to 2. And so as the volume increased, the surface area did not increase at the same rate. It was much slower. So is there any way we could adjust this so that we would still have a 6 to 1 ratio? Well, if we take that cube and we divide it into smaller cubes, well, let's then determine the surface area. So, and each one of these cubes is going to be the same size as this original one, and if we counted this up, this is 5 here, 5 here, and 5 here, so there's 25 on a layer, and there are 5 layers, so times 5, there are 125 cubes in this larger structure. Now each one of those smaller cubes has 6 faces, and each one is, the length is 1 and the width is 1. Now the volume is still 5 by 5 by 5. Okay, alright? So 1 times 1 is 1. So this is going to be 125 by 6, and then 5 times 5 times 5 is 125. So this is still, we have now gone back to a 6 to 1 surface area to volume ratio. So if you take something that has a larger volume and you divide it into smaller compartments, if you will, you're going to increase the surface area. So now you understand why you are made out of trillions of cells instead of just being one nice big cell. So the first cell we want to look at are those prokaryotic cells. We have mentioned that prokaryotic cells are smaller than eukaryotic cells, that they were the first cells to evolve, and they're pretty simple. This is an actual electron micrograph of a prokaryotic cell. If you were to look at it underneath a light microscope like we have here in the room, you would be able to make out something that kind of looks like this, and not much more than that. So an electron microscope allows you to magnify things quite a bit. And we can make out that it has like a rod shape. So now, let's look at what's inside a prokaryotic cell. Inside that prokaryotic cell, you're going to find some DNA, and that DNA is a large circle of DNA. However, it's so large that it's going to be folded, if you will. And it's in an area that's called the nucleoid region, or the nucleoid. We can also find smaller circles of DNA throughout the cytoplasm, and those are called plasmids. Plasmids may indeed also contain genes. And in addition to that, you're going to find ribosomes. I want you to take a minute and think about what ribosomes do, if you can remember from Biology 1. You should remember that ribosomes are workbenches where proteins are made. Now, outside the cytoplasm, if you will, we're going to find a plasma membrane. And the plasma membrane regulates what gets into the cell and what exits the cell. Some prokaryotic cells actually have two plasma membranes. Outside of the plasma membrane, we're going to find a cell wall. Now this cell wall is different from a plant cell wall in that it's not made out of cellulose, but a complex carbohydrate called a peptidoglycan. There are some prokaryotic cells that, in addition to the cell wall, they have a slimy capsule covering the cell wall. That slimy capsule seems to confer some extra protection for the prokaryotic cell. In addition to that, there are some prokaryotic cells that have these little plasma membrane extensions sticking out. And a singular one is called a pilus, and plural is a pili. They are used to attract another prokaryotic cell so that they can exchange DNA in a process called conjugation. There are also some prokaryotic cells that have a flagellum. And that flagellum, of course, is used for movement. However, it's a solid core of protein, and it rotates like a helicopter rotor, if you will. You may be asking yourself, how do prokaryotes obtain food? Well, some prokaryotes will decompose dead tissue, like a fallen log or some leaves. Those are called decomposing heterotrophs. If the prokaryote, instead of dead tissue, is decomposing living tissue, well, that's causing some sort of a disease, and so they're a pathogen. So there are some pathogenic heterotrophs. You may be familiar with streptococcus, which causes strep throat. And it's basically decomposing your throat tissue, causing strep throat. There are some prokaryotes that are really cool because they can do photosynthesis just like a plant. There are other prokaryotes that will do photosynthesis, but they modify it. And instead of using water, they'll use hydrogen sulfide. So some prokaryotes are heterotrophs, some prokaryotes are autotrophs. In addition to that, prokaryotes must also do cellular respiration. If oxygen will actually poison a prokaryote, and they have to stay away from oxygen, those prokaryotes are called obligate anaerobes. And the word anaerobe means without air. Obligate means that they're obligated to stay away from the air. There are some prokaryotes that can do cell respiration with oxygen, or if no oxygen is around, they can do it without oxygen. Those are called facultative anaerobes. And last but not least, there are those prokaryotes that actually embrace oxygen, and they do cell respiration with oxygen, and those are just called aerobes. Alright, so now let's look at a eukaryotic cell. Wow, my goodness! One of the things you should notice right away about that eukaryotic cell is there's lots of stuff inside. And all that stuff, most of it, are organelles that have membranes. One of the largest organelles that is so prominent is the nucleus. And then you're going to see lots of other organelles like the mitochondrion and the Golgi apparatus. So let's look then at a eukaryotic cell and a eukaryotic animal cell and a eukaryotic plant cell. Take a few minutes and think about differences between these two types of eukaryotic cells. Alright, one of the things you should have noticed right away is that they both have a large nucleus. Some differences you should have noticed is that if you're talking about a plant cell, you'll notice that this particular plant cell has a chloroplast. However, I do want to warn you, not all plant cells have chloroplast. If you are examining the tissue of a root, you're not going to find any chloroplast there. Another difference is that plant cells have a large central vacuole, or most of them do, and that large central vacuole allows the plant cell to regulate water in the cell and remain turgid, if you will. Another thing you'll notice is that plant cells have a cell wall, and that cell wall is made out of the carbohydrate cellulose. If we go back and we look at an animal cell, animal cells have this little structure right here. Oops, let's go back. Plant cells have this little structure right here called centrioles in a region called the centrosome. Plant cells have a centrosome, but they do not have centrioles. In addition to that, plant cells do not have lysosomes like animal cells do. Alright, now the question is, how did eukaryotic cells come into existence? Well, a lot of these organelles have been made from the folding in of the membrane. For instance, let's go back. If this is a prokaryotic cell, then over time what would happen is that extra membrane could fold in and end up around the DNA, if you will, and that could become the nucleus. Other organelles came into existence through the process of endosymbiosis, which we will examine later on. So as we mentioned, in order to observe most cells, you're going to have to use a microscope. And so using a microscope will allow you to magnify objects. However, sometimes when you magnify objects, you lose resolution. And resolution refers to the ability to distinguish between objects. So here we are, we're looking at some grass, and the resolution here is quite good. We can see one piece of grass from another piece of grass. We can distinguish the veins in the grass and so on. Over here we have magnified that picture 64 times, and in doing so we have lost the resolution. You can no longer see the veins in the grass, but it's been magnified. So when you are using a microscope, it's great to magnify things, but you want to make sure that you have the resolution. Sometimes you're going to need some stains in order to aid that resolution. So over here, this first picture that you're looking at underneath the microscope are some cells that have not been stained. And so you're looking at the cells with a bright field microscope, and so it's unstained. You really can't see very much. However, if you take those same cells and you stain them with something like methylene blue, now what you can see is that structure right there, and that is the nucleus. In some labs, they have what they call phase contrast microscopes. And a phase contrast microscope is one that eliminates some extraneous lights. It's a technique that converts phase shifts in light passing through a transparent specimen to brightness changes in an image. When that happens, phase shifts themselves are invisible, but they become visible when shown as brightness variations. It's kind of like contrast on a television. So what you're looking at right here is a phase contrast image, and it appears to be increase the resolution so that you can see a few more structures. Now the one right below that is very similar to the phase contrast. However, it allows you to look at images without staining them. Now fluorescence, this is a micrograph that uses fluorescence, and that kind of microscope, you're going to use a monochromatic beam of light, which means all the light has the same wavelength, and you're going to stain the specimen with the fluorescent dye. When you stain that specimen with the fluorescent dye, different structures are going to appear to have different colors. Now a confocal microscope is one in which you're using a very small point illumination of light through a pinhole, and it will eliminate out of focus light. And so what you're looking at right here is a confocal image, and below that is one that's not using a confocal microscope, and you can see the difference in resolution between the two. Now all of these images that we are looking at, the microscope is a light microscope, meaning light is used to view the image. And along then came the electron microscope. An electron microscope is a type of microscope that uses an electron beam instead of light to illuminate the specimen. And the advantage of using an electron microscope is that it increases the magnification greatly and still keeps the resolution. However, electron microscopes are extremely expensive, and you're not going to find them in most high schools. So now we're going to start examining the different organelles found in a eukaryotic cell. The most prominent organelle that you would find in a cell is the nucleus. And here's an electron micrograph of the nucleus. And here is a illustration. What is contained within the nucleus is the DNA, which has the instructions or the template on how to make all the different proteins important in regulating the cell. Quite often they'll use the analogy that the nucleus is the brains of the cell. In addition to having DNA, you're also going to find RNA and proteins that are important in protein synthesis and reproduction of the cell. In addition to that, you'll find a structure called the nucleolus, and we'll talk about the nucleolus later on, but it's a pretty prominent feature in the nucleus. And then outside, you're going to find the nuclear envelope. And it is indeed a membrane, but it actually is two membranes. And that nucleus is pitted with pores. Those pores regulate what gets into and out of the nucleus. For example, it's known that messenger RNA that's transcribed from the DNA, not all of those messenger RNAs are allowed to leave the nucleus. Only a certain percent are actually allowed out into the cytoplasm to be translated into proteins. And now what you're going to see continuous, or we say it's connected to that nuclear pore, is this beautiful structure out here called the endoplasmic reticulum, or we call it the ER. And so before we go to the ER, let me backtrack and talk about the nucleolus. We mentioned that the nucleolus is a prominent structure found in the nucleus, and it contains DNA, and that DNA codes for the production of ribosomal RNA for ribosomes. Alright, so now let's get back to the endoplasmic reticulum. I mentioned that there were two types. There's rough ER and smooth ER. Well, what's the difference between rough ER and smooth ER? Is that rough ER is pitted with little ribosomes. We mentioned before that ribosomes are the workbench where proteins are made. So what happens is, here is like a ribosome and it's making this protein, and there's a signal, if you will, on the end of that protein that will attract it to a pore on the endoplasmic reticulum. And as that protein is being made, it's going to be threaded into the inside of the ER. Later on, that protein is going to be packaged up inside a vesicle and shipped out. Usually it'll end up at the Golgi apparatus and be modified, and then it will continue on its journey out to the plasma membrane. If a ribosome is not in the process of making a protein, that ribosome will actually fall off of the ER. So if a ribosome is attached to the ER, it's actually in the process of making a protein. Alright, now connected to the rough ER, you're going to find something called smooth ER. Now, smooth ER looks a little bit different, and first off, it doesn't have any ribosomes, and secondly, it looks a little more tubular, if you will, than rough ER. You might be asking yourself, what does the smooth ER do? Well, they have found out that the smooth ER is involved in the synthesis of lipids and phospholipids. It's involved in the breakdown of glycogen. We also know that for certain drugs, it detoxifies them by adding an OH group or a hydroxyl group. By adding a hydroxyl group to the drug, it'll make it more soluble and allow it to be flushed out of the cell and out of the body, if you will. There's a specialized form of the smooth endoplasmic reticulum that's used to store reserves of calcium. Calcium is needed in order for muscles to contract. So when a muscle is going to start to contract, the sarcoplasmic reticulum will release those calcium reserves in order for the muscle to contract. Now, the next organelle is the Golgi apparatus. And the Golgi apparatus, to me, looks like a stack of pita bread, if you will, or pockets of pita bread. And there are going to be vesicles that are going to have inside proteins, and they'll come off of the endoplasmic reticulum, and they're going to be transported or attracted to the Golgi apparatus. The Golgi apparatus has two distinct faces. Where the little vesicles come in to dock, if you will, is called the sys face, or the receiving side of the Golgi apparatus. The material now is going to go from compartment to compartment. It's going to be modified. And then over here on the opposite side is called the transface, and that's the shipping face of the Golgi apparatus. So these little molecules are now going to be wrapped in little vesicles, and they're going to be shipped out to the plasma membrane, where they will be exported through the process of exocytosis. So here's a 3D illustration of the Golgi apparatus. Let's watch this little video. So the functions of the Golgi apparatus, as I mentioned, it can modify proteins that have been shipped out from the endoplasmic reticulum. It's found to be involved in the manufacturing of certain polysaccharides. And as those little vesicles join the plasma membrane, it causes the plasma membrane to increase in size or contributes to the growth of the plasma membrane. So here's a little video that shows you things entering the Golgi apparatus. Now, the Golgi apparatus also seems to be involved in the production of an organelle called the lysosome. Now, the lysosome is only found in an animal cell, or some certain protist. So let's suppose that we have a particle or a food particle that's going to enter the cell through phagocytosis. Now, this food particle is inside the vesicle, and in order for the nutrients to get out into the cytoplasm, it has to be digested. So a lysosome is an organelle that was made from the Golgi apparatus that contains hydrolytic enzymes. When it fuses with the food vacuole, it will secrete or put those hydrolytic enzymes inside that food vacuole. It will break down the food, and now the smaller molecules will be allowed to go into the cytoplasm. In addition to that, lysosomes are important in recycling cell parts. So here we have a little mitochondrion that is broken down and has been wrapped up into like a little vesicle, and so now the lysosome attaches to that vesicle, secretes in the enzyme, the components of the mitochondrion is broken down, and now those little organic molecules can be recycled. The nickname for the lysosome is called the suicide bag. You might be thinking to yourself, why is that? If you have a large number of lysosomes break open at the same time, those enzymes can start breaking down the organelles and actually kill the cell. There are times when cells are destined to die. Think about the tail of a tadpole. We know that it gets smaller, and it just doesn't fall off. The question is, how does that happen? The cells of the tail of the tadpole starts to die because these lysosomes are breaking open in a process called apoptosis. We also know that when you were a little embryo that you used to have skin between your fingers. That tissue is no longer there, and that's because of those lysosomes breaking open. The next organelle that we want to look at are vesicles and vacuoles. The difference between vesicles and vacuoles is that, to me, it's a matter of size. Think of a baggie versus a garbage bag. Vesicles are much smaller. They're membrane bound. We see them when we're talking about the Golgi apparatus and vesicles coming off of the endoplasmic reticulum. Vacuoles are much larger, and they are also membrane bound, and they are usually used for storage. For example, a water vacuole will store water. Sometimes you'll also find pigments in them or crystals in them. Plants will use a large central vacuole to help maintain their turgor and structure. There are some vacuoles that do not remain static, but they interact with the environment. Some little protists have water vacuoles that contract and will get rid of excess water. So we have a little video that shows you this process of a water vacuole. This first one is of Chlamydomonas. It's hard to see the little vacuole contracting. You have to look very carefully. Okay, this next one is a close-up of a So this leads us to a concept of something called the endomembrane, or in some books is called the cytomembrane system. And it refers to this collection of membranes that are continuous or give rise to one another. For instance, the nuclear envelope is continuous with the endoplasmic reticulum, and the rough endoplasmic reticulum is continuous with the smooth endoplasmic reticulum. The endoplasmic reticulum gives rise to the Golgi apparatus. There are little vesicles that are secreted by the endoplasmic reticulum that will come and fuse to the Golgi apparatus. The Golgi apparatus makes lysosomes. Also, these little vesicles, as we've mentioned before, as they're destined to be secreted, will fuse with the plasma membrane and contribute to the surface area of the plasma membrane. So all of this is referred to the endomembrane or cytomembrane system. Now there are membrane or bound organelles that are not a part of the endomembrane or cytomembrane system, and that includes things like microbodies, mitochondrion, and the chloroplast. So you might be asking, what in the world are microbodies? Microbodies are a collection of membrane-bound organelles that contain enzymes that function in a certain manner. For example, peroxisomes and glycosomes are microbodies. Now, a peroxisome will remove hydrogen from certain toxic substances and then combine the hydrogen with oxygen to form, guess what? Hydrogen peroxide. Now you know hydrogen peroxide is a poison. So a peroxisome will have catalase to break that hydrogen peroxide down into water and oxygen. Now, it turns out that when you drink alcohol, which contains ethanol, this is the way that ethanol is detoxified. Now, a glyoxysome is a microbody in plant cells that has the ability to convert fatty acids to sugars. Plant cells do not have glyoxysomes. And so if you're talking about a seed that has oils, they have the ability to take that oil and turn that into carbohydrates. So the last thing we want to look at in this little PowerPoint is the mitochondrion. And the mitochondrion has been called the powerhouse of the cell. And it's this wonderful little organelle that has the ability to take ADP and convert it into ATP. ATP is the molecule that is used in a lot of energy conversions within the cell. It appears that the mitochondrion used to be a prokaryotic cell, and we do have support or evidence that once upon a time this was a prokaryotic cell. First off, it has DNA, but the DNA is circular and resembles prokaryotic DNA. The mitochondrion also has ribosomes, but the ribosomes found in the mitochondrion resembles that of a prokaryotic cell. The mitochondrion also does protein synthesis, but does it like a prokaryotic cell. And so that evidence supports the fact that once a time it used to be a free-floating cell, and that it became engulfed, if you will, through the process of phagocytosis, and it just kind of took up residence and became an organelle. Now, if you examine a mitochondrion, you will notice that it has two membranes, an outer membrane that is nice and smooth, and an inner membrane that is folded. That inner membrane is called the cristae, and on the cristae you will find proteins to form something called the electron transport chain that is used in this process of making ATP. So one more time, the mitochondrion is important in that it has the ability to take a phosphate group and add it to ADP and make ATP. Alright, so that's the end of our presentation, but before we leave, I'd like to show you a 3D little video of the mitochondrion. Kinda cool. Alright, so we will see you in the next PowerPoint to talk about the rest of the structures found in a cell.